Hi students, hope all are doing well. So, we started with the next chapter of biotechnology and its applications. In this biotechnology and its application, we have already covered one main topic. What was that topic? Bt cotton, isn't it? So, in biotechnology in agriculture, the first point what we discussed was Bt cotton. Hope everyone is clear with that concept. Next very important point what we are going to move on to is developing pest resistant plant. What we are going to develop? We are going to develop a pest resistant plant, GM tobacco. What we are going to develop? GM tobacco. What is GM? I told you GM stands for genetically modified. So, this tobacco what I am going to make is a genetically modified tobacco. With the help of rDNA technology, I am making this tobacco. That is why the name of the tobacco plant is what? GM tobacco. Okay. And in this process, when you see the second application of biotechnology in agriculture after the Bt cotton, this process we are calling it as giving a specific name to the process. What is it? It is RNAi. You can find what is that process called? RNAi. Okay. So, what is this RNA? RNA, you know, ribonucleic acid. And what is this small I standing there? That is referring to the term interference. What it is referring to? It is referring to the term interference. Okay. So, RNAi, or it is the full form is what? It is RNA interference process. Okay. And this process can also be called as, we can also name it as mRNA silencing. What do we can name it? We can also name it as mRNA silencing. Okay. And what is this RNAi and what is this mRNA silencing? Actually, this is a defense mechanism which is taking place in all eukaryotes. So, you know what is an eukaryote, isn't it? So, eukaryotes are the organisms which are having a well defined nucleus with different cell organelles, membrane bound cell organelles. We call them as what? We call them as eukaryotes. Now, when you are seeing this is RNAi or mRNA silencing is a method which is helping as a defense mechanism adopted by whom? The eukaryotes. Okay. So, it is a cellular defense mechanism. Cell is defending itself. Okay. So, that is a process what we are going to discuss in detail in this today's class. Hope it is clear. So, what is the topic we are going to discuss? We are going to discuss about GM tobacco. Okay. So, tobacco is the plant which we are going to discuss. And what is the process which we are going to discuss? We are going to discuss RNAi or RNA interference. Otherwise, it is also called as what? mRNA silencing. And what is it? It is a method, a defense mechanism which is adopted in which one? The eukaryotic organisms are having this defense mechanism within themselves. Okay. So, what is the defense mechanism which is taking place? Listen students, for this you should just recollect the point which we have learnt in the last class. What was that point? We learned for any protein to produce in a cell, there are two processes involved. What are the two processes? The first process is transcription and the second process is translation, isn't it? So, this we have discussed. So, DNA, from the DNA, the gene is being converted into what? RNA. That process we call it as what? Transcription. From that RNA, that is mRNA. So, DNA to mRNA, it is transcription, and from that mRNA what is produced? The protein is produced. Whatever the DNA gene which is coding for, that type of protein is produced. So, for forming of any protein or forming of any enzyme or forming of any substances, when you are seeing, there are two main processes involved. One is transcription, that is copying of the DNA and getting converted into mRNA, and formation of protein from mRNA, we call it as what? Translation. Is it clear? You already know. Now, what is the defense mechanism then plants have? Listen carefully. What plants do is that when they are acting as a defense mechanism, what they do is that the DNA, the gene which is coding present in the DNA, that has to be converted first into what? RNA, isn't it? Instead of forming this RNA, that is mRNA, they are forming another sort of RNA which we call it as double stranded RNA, dsRNA. What they are producing? They are producing dsRNA. That is the defense mechanism. They have a special mechanism for that. That you are not going to learn at this level. In your higher studies, you will be learning the same. Okay. So, instead of producing mRNA, if a eukaryotic cell is producing dsRNA for the same, then it is a type of what? Defense mechanism. So, instead of forming mRNA, what they are producing? So, the DNA is being converted into dsRNA. How it is getting converted into? It is getting converted into dsRNA instead of mRNA. It has to form what? mRNA. Then only DNA to mRNA, mRNA to protein. Now, when this DNA is forming dsRNA, there is some mechanism taking place for defense. Okay. So, instead of producing mRNA, what they are producing? They are producing dsRNA. So, what is dsRNA? It is double stranded RNA. That is the defense mechanism which you can play. You can find that it is taking place here, right? Now, listen carefully. We are going into the story of RNA interference in tobacco, how it is playing a major role. Listen very carefully, a bit difficult to understand, so listen very carefully. In detail, I will be explaining one by one. Okay. See, this green color portion what I have drawn here, isn't it? So, this one you can see, this is a plant. What is it? It is a plant. What plant it is? Here we are going to learn about tobacco. So, this is the tobacco plant and this is the root of the plant. What is that part? It is the root of the plant. Okay. And here you can find the brown color part, right? So, what is that? That is the nematode, the worm, the pest which is going to attack the plant. Okay. So, this part is the plant and this part is the pest. Is that clear? Fine. Now, this plant, when you are seeing this plant is what? Tobacco I told you, and it is GM tobacco. GM tobacco means what? Genetically modified tobacco. So, it is not the natural tobacco, it is a genetically modified tobacco, which means here in the cells you can find a gene is present. That gene when you are seeing is coding for some toxin, and that toxin when you are seeing, what they are doing is that if the pest comes and attack this plant, the pest itself gets killed. Okay, the pest cannot survive. So, for that some rDNA should be there, isn't it? Some foreign gene should be there inside the plant. Then only this is possible for making this GM plant, yes or no? Yes. So, for making this GM tobacco, I am introducing what? I am introducing a gene into the tobacco plant, right? How can I introduce? In plants we have already learnt, I am using a vector. And what is the vector here? The vector what I am going to use here is Agrobacterium tumefacium. What is the vector what we are going to use here? We are going to use the bacterium Agrobacterium tumefacium, right? So, this is the GM tobacco what I am going to make. For making that, I am introducing a gene of interest, isn't it? So, a DNA, the gene of interest, I am introducing it with the help of what? A bacteria named as what? Agrobacterium tumefacium. The gene is being introduced into this plant, right? Till now it's clear? Fine. So, tobacco is the plant. Now, who is the pest? I should know who is the pest, isn't it? The pest is attacking. So, I should know something about the pest. The pest is a nematode. What is the pest? It is a nematode. Nematoda you have learned, isn't it? Like you have learned in your invertebrates category, right? Many categories were there: Porifera, Coelenterata, Ctenophora, Platyhelminthes, Annelida, Arthropoda, Nematoda, and all you have learned, isn't it? So, in that nematoda, roundworms. Who are they? The roundworms. They are the one who is acting as a pest in this tobacco. They are going and attacking the roots. They are eating up the plant, the roots, and thereafter they are causing harm to the plant, right? So, this nematode is name of the nematode. The binomial name of the nematode is Meloidigne incognitia. Okay, so Meloidigne incognitia is the name of this nematode. which is attacking what? This tobacco plant. So, this is the nematode which is attacking the tobacco plant. Is that clear? So, Rna incognitia is the pest. What it is? It is a nematode. It is a roundworm which is attacking the plant. Now, listen carefully. We are moving into the topic, okay, in detail. The first thing is that gene is introduced into the cell, isn't it? So, in the cells of these plant, one gene is introduced with the help of Agrobacterium tumefacium. The Agrobacterium tumefacium is a pathogenic, means they induce tumor, isn't it? So, they are getting in and they are spreading in the roots of the tobacco plant. Now, when you are seeing in that, the foreign gene is also present. The gene which is coding for some substance is also present. So, this gene, when it is introduced into this plant, the plant is a eukaryote, isn't it? So, what this plant is doing is that the defense mechanism I have told. Usually, if you are introducing a gene, the DNA has to be getting converted into what? It has to get converted into mRNA, and this mRNA should produce the protein, isn't it? But here some defense mechanism is taking place. So, what happens? The gene, after it is introduced into it, the gene, when you are seeing, they are producing by some defense mechanism. What they are producing is that they are producing a DNA, okay, dsRNA. What they are producing? They are producing dsRNA. So, what is dsRNA? RNA means only one strand, copying will be done. But when you are seeing here, this dsRNA means double stranded, means the sense and antisense both they are producing. Like a DNA, they are having what? Complementary base pair. So, here A means here T will be there, here C means here G will be like that they are producing what? Double stranded, not DNA, they are producing what? Double stranded RNA. So, this mechanism is a defense mechanism, and this mechanism is somewhat different, which you need not learn, okay. So, in this actually what is happening? Instead of producing from the gene, the DNA is not producing mRNA, instead they are producing what? dsRNA, clear? So, the gene I have introduced with the Agrobacterium tumefacium, this Agrobacterium tumefacium, after entering into the DNA, when they are making different substances, coding for different characters, one of the gene what I have introduced is making what? They are making dsRNA. What they are making? They are making dsRNA, double stranded RNA, clear? Now, this double stranded RNA is being acted upon by an enzyme. Name of the enzyme is dicer. What is the name of the enzyme? Dicer. So, this dicer is digesting this double stranded RNA into many small fragments, okay. So, they are being digested into what? Like this, here you can find 3 fragments, the dsRNA is being digested, right, and you can find small fragments are present. So, that small fragments, I call it as small interfering RNA. What do you call it? It is called as small interfering RNA, or we call it as siRNA. What do we call them? We call it as small interfering RNA or siRNA, okay. So, siRNA. So, what is si? Small interfering RNA, okay. So, one dsRNA was formed. This dsRNA is broken down into small fragments. These small fragments we call it as what? Small interfering RNA. Is that clear? Till this it is fine, right? Now, listen very carefully. Here this nematode is going to come and attack. So, the small interfering RNA is present. This nematode is going to come and attack the plant, okay. Whenever the nematode is getting into the plant, at that time you can find in the plant there is a protein complex present. The name of the protein we call it as what? Argonoid protein. What do we call them? We call them as, giving a name, argonoid protein. What do we call? Argonoid protein, okay. What this protein do is that, you know, I told you small interfering RNAs are present, right. These small interfering RNA, when you are seeing the small interfering RNA, so this is the small interfering RNAs which are present. So, this is one big in that small. This protein goes and binds with this small interfering RNA. Protein is going and binding with what? The small interfering RNA. What is that protein? Organoid protein. Okay. They go and bind. Thereafter, what they do is that they digest. What are they doing? They are digesting. Digesting means, you can understand, they are eating up. So, this is the RNA which was present, dsRNA, isn't it? Small fragments. So, this dsRNA, when you are seeing one strand, so this strand is digested. Digested means what? Eaten up, right? And only single strand is present, that one of the strand has been digested by whom? This organoid protein complex, right? So, I repeat again. So, till now what we have discussed, a bit difficult to understand, students, that is why I am discussing in detail. You can find a gene is introduced into tobacco plant with the help of a bacterium, Agrobacterium tumefacium. This DNA gene, after getting into with the help of this plasmid, Agrobacterium tumefacium, what is happening? Instead of producing mRNA, they are producing dsRNA. This dsRNA, in the presence of an enzyme dicer, it has been broken down into many small fragments. These small fragments we call it as small interfering RNAs. These small interfering RNAs, when you are seeing when the nematode is coming and attacking, the small interfering RNAs are being converted into single stranded, means digesting. One strand is being digested of this double stranded RNA. The siRNA, one strand is being digested with the help of a protein called as what? Organoid protein. Okay. So, this complex which is formed, I told you, the what is that? Your small interfering RNA as well as your organoid protein, when they combine together, they are producing a protein complex. What they are producing? They are producing a protein complex where they are helping in digestion of one of these strands of these siRNA, isn't it? So, that complex what they are producing, we call it as RNA induced silencing complex. What do you call that? RNA induced silencing complex. Otherwise, in short we write it as RISC, RNA induced silencing complex, RISC. Clear? Now listen carefully. In this nematode also there is a gene. In this nematode also there is a DNA. That DNA is also having the gene. Listen very carefully, very important. That DNA is also having a gene which is responsible for the growth of the plant. Okay. So, I am telling, so imagine this is a nematode. Okay. This is a nematode. The nematode is made up of cells. In each and every cells, when you are seeing, what is present? DNA is present. In that DNA, there is a gene which is used for the growth of the plant, right? Means if the, sorry, not plant, the nematode. So, if that gene is present, then only the growth factor, the protein will be produced, which will be helping in the growth of the plant, right? So, in this nematode, usually what happens is that the DNA will be converted into mRNA. Okay, what is that process called? We call it as transcription, isn't it? From that mRNA, what will be produced? This protein is produced. So, there is a DNA, in that there is a gene. That DNA gene, when you are seeing, they are getting converted into what? mRNA. mRNA is getting converted into protein, and this protein coding gene, the gene which is coding for this protein, if this protein is made, only then the nematode can survive. Okay. If this protein is not there, means if this protein metabolism is not taking place, protein production is not taking place, then what will not happen? You cannot find that nematode is growing, they cannot grow. Okay. So, DNA to mRNA transcription, mRNA to protein, we call it as what? Translation. Usually in the nematode this is taking place, but here what happens? Listen very carefully. Okay, here what is going to happen? So, this is the nematode, isn't it? So, it is having its DNA. This DNA, by transcription, what they are producing? They are producing this mRNA. Now, this nematode has got into the plant. In the plant, so outside DNA to mRNA, mRNA to protein, nematode is growing. But when this DNA means nematode has got into the plant ke bhitar chala gaya hai, so they has gone into the plant. Now, this nematode DNA will produce mRNA. Okay. but this mRNA, when you are seeing, will be silenced, means transcription will take place. DNA to mRNA is taking place. So, DNA to mRNA is taking place, transcription is taking place. But this mRNA, when you are seeing, when it is being produced here, this mRNA is being, so I told you here one of the strands will be digested, isn't it? So, here what is happening? One of the strand is getting digested, siRNAs, one strand is getting digested, only one strand is present. So, here you can see only one strand is present, isn't it? So, that strand and this strand is complementary to one another. Complementary means what? This strand as well as this strand, when you are seeing, they are complementary, means there A means here T is there, there G means here C will be there, like that they are complementary to one another. And this mRNA which is present over here in the plant, okay, what they are synthesizing, they will go and join with this mRNA of the which one? The nematode. So, this siRNA which was there is going and connecting with whom? This mRNA. Now, this mRNA is getting joined with this one, okay, they are binding with one another. So, when they are binding with one another, what do you think? Whether the mRNA will be expressing and forming the protein? No. Why? Because this mRNA which was produced is going to produce protein, but this mRNA when it was produced immediately complementary. This siRNA which was present is going and attaching with this mRNA. So, now they are binding with each other and the translation process is stopped. So, what is the process stopped there? You can find what is not taking place. Translation is not taking place. The mRNA is silenced. Silenced means what? The mRNA is suppressed. The mRNA's role is compressed. No, you cannot do. This one is going and attaching to this mRNA and the mRNA is getting silenced, the mRNA is getting suppressed and the translation process is not taking place. If the translation is not taking place, then what will not be produced? The protein will not be produced. If the protein is not produced, do you think that nematode will be growing in the plant? No, they will die, okay, because of which the nematode is dying. So, like that, I am making a pest resistant plant. Clear? So, this is regarding RNAi or RNA interference, okay. So, in the text what and all terms are used, most probably it has been dealt. Other than that also extra information is given. Once more I will repeat. Before that I have to tell you something, some more points. What has to be discussed is that, listen carefully what has been missed out here, okay. So, what has been missed out? One is your source of the DNA. So, this DNA I am introducing is into the plant, isn't it? I told you one gene I am introducing it into the plant with the help of a bacterium called as what? Agrobacterium tumefacium, okay. So, this gene which I am introducing into the plant for making this GM tobacco, this gene where from I am getting? What is the source? Source means kaha se mil raha hai, okay. So, where from I am getting it? I am getting it, the source can be two. One is I am making it or I am getting it from two sources. One is the transposomes or the jumping genes or mobile elements. So, there are some sets of genes, we call it as what? The jumping genes, which jumps from one place to another. Much in detail don't go into. So, these names we call it as what? The jumping genes, okay. So, mobile elements or jumping genes, from there I am taking this gene, or we are taking it from retrovirus. RNA viruses I told you, RNA which is capable of making DNA. You have learned retrovirus HIV. HIV after getting into a body, what will they do? They will get, with the help of the enzyme reverse transcriptase, RNA will be converted into DNA. So, like that also this gene is possible to get, okay. So, we can get it with the help of what? Retrovirus. Retrovirus, when you are seeing the RNA, will make their complementary, okay, they will make their complementary base pairing and they will get converted into what? DNA, cDNA, complementary DNA we call, okay. So, this is also possible. So, these are the two sources from which I can get the gene to put it into what? This plant. So, what are the two sources? One is the jumping genes, or we call it as the mobile elements, or we call it as transposomes, okay. All are one and the same. From there we can introduce, or we can introduce with the help of what? RNA viruses, the retrovirus. The category we call it as what? The retrovirus. So what is the question which is asked? The question is asked, so where from this gene you are introducing into? What is the source of the gene? The source is two. One is transposomes or RNA viruses. Clear? And this gene, with the help of what vector you are introducing? Agrobacterium tumefacium. This, after getting into this gene, the gene actually what it has to do? It has to produce mRNA and thereafter it has to produce the protein. But the gene, after entering, what they are doing? Cellular mechanism, defense mechanism. They are converted into dsRNA. This dsRNA is broken down into many fragments, okay, small interfering RNA. With the help of an enzyme, dicer. This small interfering RNA, in the presence of a protein, arachnoid protein, they are getting a complex. They are forming a complex called as RNA induced silencing complex, which will break one of the strand. RNA's one strand will be broken, okay, and that strand, when you are seeing, one single strand will be present, and this single strand will be complementary to which strand? This mRNA. So this single strand RNA will go and bind with which one? This mRNA. And in the nematode, the same mechanism, they are also having the DNA. The DNA is producing a protein for the formation of what? Growth of the nematode. But when you are seeing this DNA transcription, they are producing mRNA, but the mRNA is getting silenced by this. When this nematode is entering into the plant, this segment is going and complementary to this mRNA sequence, they are going and attaching with this, and that mRNA is silenced and the mRNA is not able to express and the mRNA is not producing protein. When the mRNA is not producing protein, you can find the growth is not possible in the plant, they die. Like that, you can find we are developing what? Pest resistant plant. Okay. So this is regarding your RNAi or RNA interference. Hope so it's clear. We will be discussing the writing part of the same. With this diagram, to some extent it's clear, I think so, right? Now we will be discussing about the writing of the same. Clear? Okay students, so let us discuss what is given in the book, line by line. If you are read on, you will understand what it is. Okay. Now moving on to the pest resistant plants which we have discussed, we are going to produce what? We are going to produce GM tobacco, isn't it? So for producing this GM tobacco, what we are going to do? See, a nematode, Melodygnia incognitia, infects the roots of tobacco plant. I told you that is the nematode roundworm which is attacking the tobacco plant, and it is causing great reduction in the yield, means the tobacco yield is becoming less. So business, when you are saying, it's coming down. A novel strategy was adopted to prevent this infestation. So what is that strategy? That is RNA interference or RNAi control strategy. Okay. So RNAi takes place in all eukaryotic organism as a method of what? Cellular defense. I told you it is a defense mechanism taking place in eukaryotes. In this method, actually it is involving silencing of the specific mRNA due to complementary dsRNA. Okay. So complementary dsRNA is going and silencing whom? This mRNA that binds to and prevents translation. What is not taking place? Translation is not taking place, protein is not formed. So this is called as what? RNA silencing. What do we call them? They call it as RNA silencing. So what is RNA silencing means? You have to explain this one. This is the definition. Okay. The source of this complementary RNA, okay, from where I am getting this complementary RNA, it is produced in the plant, okay, but from where I am getting it? I am getting it by infection, okay. So it can be a RNA genome, I told you, it can be an RNA genome, the retrovirus, or it can be mobile genetic elements, we call it as what? The transposomes, okay. So these are the one which are acting as an intermediate, helping in transferring the gene from with the help of a vector, Agrobacterium tumefacium, it is introduced into the plant. So what is the source of these complementary RNA, cRNA? So this complementary RNA, we are getting it from where? dsRNA. For making that the gene, we are getting it from mobile elements, or we are getting it from what? RNA genome like retroviruses. Clear? Next, using agrobacterium vector, this nematode specific genes are introduced into the host plant. I told you, with the help of agrobacterium, this gene which we are getting it from the transposomes or from the retrovirus, I am introducing where? I am introducing it into the tobacco plant with the help of a vector agrobacterium tumefacium, right. The introduction of this DNA, when you are saying when this is being introduced, they are producing dsRNA. DS means what? They are having both the strands, sense as well as anti-sense, which are complementary to one another. They are producing double strand instead of one strand, which usually DNA produce mRNA, one single strand. Here double strand, dsRNA will be formed. One is sense and another one is anti-sense. So, two strands are produced. Okay. So, that is why the name dsRNA. Next, these two RNA being complementary to each other, we call it as what? dsRNA. That initiated RNAi. So, this is one who is going to initiate the process RNA interference. Okay. So, that mRNA of the nematode is getting silenced. Clear? The sequence was that the parasite could not survive in the transgenic host, expressing the specific interfering RNA. I told you, they produce what? They produce specific interfering RNA. Okay. So, which is going to help in what? Breaking, going to go and silence what? The mRNA. The transgenic plant therefore got itself protected from the parasite. So, this is what is happening in case of what? The GM tobacco. Hope it is clear. So, this is the diagrammatic representation how the nematode is getting into the root of the tobacco plant. Okay. With this, I am winding up for today's portion. It's hope so it's very clear, right. So, I will just show you the diagram which we have already seen in detail regarding the same. So, here you can find, I will repeat once again, transposomes or RNA viruses. With the help of that, the gene is being introduced into the tobacco plant. The gene, after going into, instead of producing mRNA, they are producing dsRNA, which is getting broken down into fragments. That fragments, when you are seeing one protein, arachnoid protein is going and forming a complex, and one of the strand of the dsRNA is broken and one strand is present. And here in the nematode, when it is getting into, in that also you can find the nematode is having the DNA. The DNA, usually that nematode will produce what? mRNA, general process, but this mRNA, when you are seeing, it's complementary base pair, since it is present here already, this plant is making, this one is going and attaching with this mRNA, and the mRNA is getting silenced, protein is not formed. If the protein is not formed, the nematode cannot grow here, the nematode will die. So, that is how the RNA interference is taking place. Hope so it's clear. That's all for today. Thank you.